Welcome to the microbiology presentation for Step 2CK. We're going to learn all about the gram-positive bacteria today, and it's going to cover everything you could possibly imagine about this topic. And you'll see what I mean when we get started. I'm Dr. Iyer with Extreme USMLE Coaching. Let's get started. So we're going to start first with a flowchart of all the gram-positive cocci. So it's very hard to differentiate all these different gram-positive cocci organisms. So I'm going to give you a nice smooth flowchart that'll help you figure this out. So we start with a purple gram stain. So gram-positive is defined as a purple gram stain, and all gram-positive cocci are facultative anaerobes, meaning that they can live in anaerobic environments and also in aerobic environments as well. So a gram stain being positive means it will stain purple or blue, and a gram stain being negative means it will stain red or pink. A positive gram stain means that the peptidoglycan in the cell wall of the bacteria picks up the crystoviolet stain, creating a purplish blue color. And a negative gram stain means that you'll have no peptidoglycan on the surface picking up the stain. And the reason for that is that peptidoglycan in gram-negative bacteria is found underneath the outer membrane, so it won't have access to the crystoviolet stain. So the first way to differentiate these gram-positive cocci is catalase. If it's catalase positive, we're going to have our Staphylococcus. So this means that they're in clusters, or they look like a bundle of grapes, and this will be very important later for identification. If it's negative catalase, it's going to be Streptococcus, and Streptococcus is found in chains. This is our major distinctive classification. And we'll keep going. After we know that it's catalase positive, we need to see what happens with coagulase. So if it's coagulase positive, it will be our favorite Staph aureus. And if it's coagulase negative, we've got some options here, and we use novobiocin to differentiate them. If the bacteria is sensitive to novobiocin, it'll be Staphylococcus epidermidis. And if it's resistant to novobiocin, it'll be Staphylococcus saprophyticus. And we're going to go over all these in more detail later, but these are good to know for now. For Streptococcus, we'll talk about how they hemolyze blood in agar. And if it's alpha hemolytic, that means it just mildly or partially hemolyzes blood. And this green color that you see in this blood agar is the partial hemolysis of blood. But if you notice, there's no clearing around it. Because there's no clearing around it, there hasn't been any complete hemolysis yet. There's still blood present surrounding the bacteria. So it's called alpha hemolytic or partial hemolysis. And to further differentiate them, we need to use something called opticin. And this will tell us which bacteria, which type of Streptococcus alpha hemolytic it is. So if it's sensitive to opticin, it'll be Streptococcus pneumoniae. And if it's resistant to opticin, it'll be Strep viridans. And Strep viridans includes Streptococcus mutans and mitis, and these are found in your mouth and cause dental plaques and caries. Now beta hemolytic. This is the classic one you always hear. And beta hemolytic means you have complete hemolysis of blood. And a picture here is that you see in these bacterial colonies, there's a circular empty clearing around it. See these empty spaces? Those are areas of complete hemolysis that you do not see here. That's what beta hemolytic means. And to further differentiate them, we have bacitracin. If the patient is the bacteria is sensitive to bacitracin, it will be group A strep, Strep pyogenes. And if it's resistant to bacitracin, it will be group B strep, or Strep agalactiae. Now we have gamma hemolytic, which means that there's no hemolysis of blood at all, not even partial. And to differentiate them further, we can see how well they grow on 6.5% sodium chloride. If there's growth, it'll be a very tough species, that's Enterococcus. And if it's no growth, it'll be Streptococcus bovis. So this flowchart is actually very important to know to be able to differentiate these bacteria, and it will help you in the future when you want to figure out what's causing your infection. Alright, let's talk about Staphylococcus aureus, our first bacteria here. There's some important points we want to go over, okay? The first thing, like we mentioned before, it's a gram-positive cocci. Cocci means in a circle, in clusters, and commonly colonize the anterior nares. So MRSA and regular Staphylococcus aureus can colonize the nares, and that's where they stay for a prolonged course. They're also encapsulated. They have a polysaccharide capsule. So this is very important. The polysaccharide capsule is going to play an important role in the bacteria's immunogenicity, or pathogenicity, because it allows for evasion of phagocytosis. So the fact that the Staphylococcus aureus is encapsulated makes it more pathogenic because it can cause more infections because it evades phagocytosis by the macrophages and other cells. So we have catalase positive. That means it has the ability to convert hydrogen peroxide into oxygen and water. So it's catalase positive, very important to know. That was the first distinguishing criteria. So what does this mean? This is going to cause a lot of problems in patients with chronic granulomatous disease. And now we're going to find out why. So in patients with chronic granulomatous disease, we have a problem with the NADPH oxidase. So let's first talk about this pathway. We have oxygen here that every aerobic bacteria and human has to deal with, and oxygen can become radicals very easily. We have an enzyme called NADPH oxidase that converts oxygen to superoxide radical. Then superoxide radical encounters superoxide dismutase to make hydrogen peroxide, and hydrogen peroxide can go under two pathways. It can go to catalase and become H2O and O2, or it can undergo myeloperoxidase to become HOCl, which is bleach that kills bacteria. So our body has different ways to make hydrogen peroxide. Bacteria are normally making hydrogen peroxide in large amounts because they encounter oxygen as well. So they can make hydrogen peroxide, and so can NADPH oxidase make hydrogen peroxide. In chronic granulomatous disease, though, we have a loss of the NADPH oxidase. So we don't have NADPH oxidase, we lose this whole part of the pathway. Alright? But we still can make some H2O2 because we have bacteria present in the body which make hydrogen peroxide, and this can get converted by myeloperoxidase to bleach, and you can still have bacterial killing in the body. But here's the problem though. What if the bacteria have catalase? If the bacteria have catalase, the catalase is going to break down the hydrogen peroxide into H2O and O2, and you no longer have any way of making bleach to kill the bacteria, so the bacteria start proliferating. So this is why in chronic granulomatous disease, catalase-positive bacteria become a major problem because they break down all the H2O2 that's inside the cells, which is made by the bacteria, and you cannot make the bleach that kills those bacteria, so they start proliferating like crazy. So some good catalase-positive organisms to know are Candida, Aspergillus, Pseudomonas, and Staphylococcus aureus. Okay, let's go back to our presentation now. This is why it's problematic in CGD. Another thing is that Staphylococcus aureus is coagulase positive. So it coagulates. It converts fibrinogen into fibrin, which is part of the coagulation pathway cascade. And this forms clots and coagulates. And we'll talk about what this means. It creates an insoluble fibrin coating surrounding the bacteria. And this is going to promote abscess formation because this big clot surrounding the bacteria actually protects the bacteria. So you have protection from phagocytosis. And that'll make them live longer inside of your body, which is good for them, bad for us. MRSA is resistant to beta-lactams. This is a major disease that's been spreading really rapidly of recent times. And the reason why is because it's got this MEC-A chromosomal gene. It's resistant to beta-lactam. It's called methicillin resistant. Methicillin is a type of beta-lactam. And what happens? MEC-A is a chromosomal gene that codes for penicillin binding protein 2A. This is different than the typical penicillin binding proteins that beta-lactams interfere with. And because it's a mutated form of penicillin binding protein, it doesn't bind to beta-lactams, rendering them useless. So spread of MRSA can actually be prevented. And this is by good hand washing and fomite surface disinfection, especially in hospitals. MRSA is a very major nosocomial infection. Nosocomial meaning happening in hospitals. Now we have some toxins to talk about. And all three of these toxins are superantigens. So what exactly is a superantigen? Sounds like a pretty cool name, huh? So a superantigen means that this antigen, or toxin, will bind to both the MHC2 on an antigen-presenting cell like a macrophage, and also binds to the T-cell receptor on a CD4 T-cell, and it will hyperactivate both of these. So it will cause an activation of the T-cell, and it will also cause activation of the macrophage. And by doing that, you get a massive cytokine release and major problems in the body. And we'll talk about that as we go on. Remember, cytokines are the reason you get fever, the reason you get hypotension, the reason you get a lot of problems associated with disease. So you have exfoliative toxins. This is a type of superantigen. So exfoliative, remember exfoliate means to lose hair or lose something. So you're going to lose some skin in this case. It's a serine protease that targets desmoglein 1. So desmoglein 1 is a desmosome holding together the stratum granulosum. So if you can't hold together a part of the epidermis, it's going to fall apart. And that's what's going to happen. So you're going to have an exfoliation of the stratum granulosum. And this is involved in staphylococcal scalded skin syndrome, which we'll get into shortly. And a key feature of staphylococcal scalded skin syndrome is there's actually no leukocytes or bacteria in the affected epidermis or in the actual bullae vesicles that form. And the reason why is it's all because of the toxin. The toxin causes everything. The leukocytes and bacteria have nothing to do with it. And the body generates antibodies to the toxin, and this is how you resolve the infection. Alright? Now we have enterotoxins. So enterotoxins are an acute onset food poisoning that occurs within 6 hours of eating the food, and we'll talk about that shortly as well, and usually resolves pretty rapidly also. A key feature of this enterotoxin, though, is that it's very heat stable. It can survive cooking, and it also can survive the gastric acid and jejunal enzymes and can cause major problems. Toxic shock syndrome toxin is associated with tampons and nasal packing. So this is different from streptococcus. Staphylococcus is associated with tampon use, so prolonged tampons, and nasal packing, and the bacteria grow and start producing this toxin. And this toxin is really dangerous because it can cross mucosal barriers and spread everywhere, triggering a massive immune response. And the enterotoxins, by the way, are only severe for those 24 hours, but then they resolve on their own. Now let's talk about a quick sample question. So, which of the following is the best method of preventing this outbreak? So remember, we always want to read the question first so we know what we're looking for. Longer cooking time, refrigeration of foods, earlier preparation, prophylactic antibiotics, or hand washing. So we want best method of preventing this outbreak. So we need to figure out what this outbreak is and how best to prevent it. So we have 8 adult men and women present to the emergency department with symptoms of nausea, vomiting, and diarrhea. All of the adults attended an outdoor cookout together earlier that day. Foods available for the cookout were burgers, tacos, potato salad, and custard. Potato salad and custard were prepared the previous day. The patients are all stabilized with IV fluids and antiemetics, and they're admitted to the ward and symptoms resolved the next day. So symptoms resolved the next day. They happen after a cookout. Potato salad and custard are involved, and it was earlier that day, so it happened pretty rapidly. So, and they were also prepared the previous day. So these are all risk factors for staphylococcal enterotoxin food poisoning. So the reasons why is that you had potato salad and custard being made, which are high-risk foods, which we'll talk about later. These symptoms were pretty rapid onset. The potato salad and custard were prepared the previous day. So there was a delay between the preparation and the consumption, which could allow for the bacteria to grow, and it resolved pretty rapidly with fluids and antiemetics. So how are we going to prevent this outbreak? Longer cooking time? Remember, we talked about how it's a heat-stable toxin, so it will not go away just because you cook the food longer. Next, refrigeration of the foods. So refrigeration of the foods would actually delay the toxin production and delay the bacteria from growing, so we can hold on to that one. What if we prepared it earlier, even before the previous day? That would be terrible because you'd have even longer amounts of time between the preparation and the consumption, allowing for more bacterial growth and toxin production. Prophylactic antibiotics. Would this be helpful? No, because the body is not being affected by the bacteria itself. It's being affected by the enterotoxin. And no amount of antibiotics can prevent you from having an enterotoxin or protect you from the enterotoxin. Last, hand washing. So hand washing is always very important in the hospital and in general as a whole. But is hand washing going to help in this situation? No, it's not. Hand washing is not the cause of the bacteria growing. The bacteria are growing because this potato salad and custard were kept outside, and Staphylococcus aureus is found everywhere in the environment. And also, commonly Staph aureus is colonizing the nares, so it's not usually on your hands. So sometimes you can get the custard and potato salad being super infected by a cough or a sneeze, but hand washing would not actually be effective here. So the best way to prevent it would be refrigeration of the foods, which would prevent the outbreak from happening by delaying the toxin production and bacterial growth.